So I think we've done a great job. I think the industry was, in a way, always pushed ahead by the biology, and so we were forced down a route of doing very manual processes early days. The biology was there before the engineering and the manufacturing had really caught up, and arguably that's still the case today. But we are starting to see the early signs of systems I think can start to make an impact. I think Militania with Prodigy were a long way ahead of many other people in actually producing that system and placing that. We're seeing things like Cocoon that Lonza now has, which is doing first in human studies. So we're beginning to see those early signs of automation, as we call it. And I think automation's a bit of a risky word in this context. It means a lot of things to a lot of different people. What we're really talking about there is removing the amount of manual labor that's involved. And that's incredibly powerful because it has the potential to bring down cost, which is part of patient access. But it also is vital in ensuring that we reduce risk, we reduce errors, and we make sure these are safe therapies. And that's always a problem in any industry as it begins to expand, as it begins to increase, as we do more of this. Obviously, the safety becomes really, really important, and making sure we can keep that same safety as we scale, and particularly as we scale out, because we can't just go bigger here, we can't just scale up. So as we scale out, making sure that we keep those same levels of safety and of quality of the product is what's going to be really important. So I think that almost splits into two for me. I think near term, we're seeing these more automated processes starting to arrive. And as we do more of that, I think the reliability is going to be tested as we do it, move from sort of tens and hundreds to thousands and 10,000s. We all hope we do because of the impact that can have on patients. We're going to test the reliability and those robustness of those systems so much more than we do today. And to do that, therefore, we're going to have to look at that really carefully, look at lessons we can learn from other industries, things like medical devices, and make sure that we're applying that same level of rigor to this as we would to that, because it is so different to what we do in traditional bioprocess, where we'd have just one large tank perhaps, and that has got a very different risk and reliability profile to 1,000 manufacturing systems all operating alongside each other in a factory. And then as we look longer term, I think it's looking at, have we actually got the right technologies? So of course, all the technologies we're using today are ones we've had available from a legacy point of view. They were never developed for this industry because this industry didn't exist. And so what we need to start thinking about now is what technologies does this industry need that we didn't need before? So because we're no longer working in batches of 1,000 litres, 2,000 litres, we're now working in batches of 1 litre or 500 millilitres, or maybe even one day 10, 20 millilitres, we could hope if we get the potency there. We're starting to think, I think, about the cells as individual entities rather than as a bulk, and starting to look at cell by cell type manufacturing processes rather than bulk manufacturing processes. And I think that's where we like to see longer term some of the real big breakthroughs and the transformations coming from. I think it's really hard at the moment because it is a new industry. I think what we see is just knowing what equipment is needed, how to design it, because it is a moving target, because it's not a mature industry where everyone's very confident in what they need. We're trying to develop manufacturing equipment whilst people are taking things through phase 1, phase 3 trials. And each of those therapies are quite often subtly different, but different in important ways. And each of them are uncertain whether or not they'll even make it through those trials. And so alongside that, we've got equipment providers, technology providers, trying to establish what the specification, what the needs of the equipment are likely to be. And so that's inevitably why it sometimes does feel, I think, the equipment and the manufacturing is playing catch up to the biology, because in reality it is. It is trying to track this target as we go through the phases of development, as we're deploying things in process development environments, trying to work out exactly what is needed with regard to what is the best level of automation, how much integration should we have? It's one of the great questions is, should one step be one module, or should we have 1, 2, 3 processes all combined into a single module? It creates a very different piece of equipment. The latter results in much less user interaction because there's not a transfer between processes. But if we have individual modules, we've got a lot more flexibility. And then particularly because some of the steps are much longer than others, you end up with very poor capital utilization. So if you've got one step that you're in for a few hours, another step that you're in for several days, as you do end up with say, for example, cell selection and cell expansion, integrating those two together, your cell selection equipment is only being used for a tiny, tiny portion of its time. But at the cost, if we separate that out, of an extra transfer step. So these sorts of things are evolving on the fly. The industry is learning as we go. And all the time, of course, therapies are moving through the clinical studies and wanting to be produced, because ultimately, as soon as we're through even the early studies, the phase 1, phase 2, people are looking ahead and start to ask that question: how am I going to make this? Am I going to be ready? And making sure that we are tracking that, we are moving as quickly as we can, is really important, but very, very hard, because this is an industry that is being born and evolving before our eyes. Technologies from a really diverse range of fields, some more obvious than others, sort of hospital cell processing equipment, one of the famous ones being Fresenius's Lovo system, which is one of the most interesting cell separators, certainly for physicists. It's got some very interesting fluid dynamics involved in that one. But then also things that were originally designed for bioreactors for space, being redeployed into this field. It's been a field that has drawn on all sorts of other things that no one really expected would ever be used in this context. I think that's done a really good job of plugging some gaps, actually. In the absence of those, I think we would have really been struggling. We would have been lots more human interactions, lots more tea flasks, lots more transfers, really back to the Dendrion days and the challenges that they had in quite a complicated, very manual manufacturing process. So we've been quite fortunate that those things have become available at the time this field has been emerging. And I think we may even still see more of that yet. I think as the field continues to move forwards, I think the technologies will pull from other areas, will continue. Things like analytics, I think still remains a big unmet need in both bioprocess more broadly, but specifically cell therapy that has a very particular challenge. You can't sample in such a big volume quite often, because in doing so, you're reducing this 1 litre product down in volume every time you take that sample. It's much easier to do when you've got 1,000 litres to work with. And so we are beginning to see the start of people bringing other technologies into cell therapy from other analytics environments. But that in itself is a particularly hard one, because what is it that people want to know when you're manufacturing a cell therapy? Again, we are doing a process that is unusual, and that is per individual patient in the case of autologous. So really trying to get a handle on that variability is so tricky. And so analytic technologies can help us a lot in trying to know what's going on and seeing where we do have those variabilities. But it's going to take a little while yet, I think, before we actually necessarily know what to do with all that data. It's like what we see with all sorts of companies from Google to Apple today. They're gathering the data with a view to knowing there's value in it, but figuring out what that value is later. I think we've got a similar analogy in this industry today. We want all the data, we'll collect the data, we'll find new tools to learn more about what's going on in the culture, whether it's real-time readouts of cell phenotype, cell viability, things that are quite tricky to do today without sampling and offline analysis. If we can start to learn that, we may well be able to learn more about the product we're making, and perhaps hopefully start to reduce some of the variability that we see, which is still a major pain point. Kymriah in particular has seen that with some of their manufacturing challenges, and the more we can bring down that variability, the more we can get more product hitting specification once we get down to release, and back to patient access. That's a great thing for the patients. I think that's a really interesting point, is traditional pharma, I think, had a bit of a love-hate relationship with new technology, and rightly so, actually. I think they see the value of the new technology, but it's quite difficult to adopt because, of course, adopting any new technology potentially puts at risk that massively expensive development you've just undertaken. So to bring in something new to it that might make an improvement, unless that improvement is a complete revolution, it's quite difficult to take that risk. In cell therapy, you've got a very different context. You've got very few rules that have currently been written. And so therefore you've got a lot more opportunity to bring new things in. You almost have to, because as we've discussed, the equipment doesn't necessarily exist as we sit here today. So yes, we do certainly see a lot more appetite to try things. I think there's still a challenge to adoption, because part of that is even once you've tried a new technology, how far away from being ready to go is that technology? So I think it's a maturity question. And one thing that we find we end up working on a lot is trying to bridge that gap between a new technology, a new idea that has shown some initial performance, but actually make that into a robust, reliable system that someone is happy to integrate into a manufacturing process for a clinical product. That's a big step. And similarly, they still have to keep an eye to that. The cell therapy companies need to be thinking about what risk am I taking by introducing a new technology into this, and making sure that they are confident the technology that they're trialing is actually something that can be brought into their final manufacturing process. That's one of the huge challenges in the industry today, is the costs. With Zolgensma, you've got a therapy that's $2 million, a sticker price. Even something like a Kymriah and a Yescarta, you're talking best part of 500 million. And that's before you've actually factored in the cost of the hospital and the likes, which potentially push you up near $1 million. So the costs of these are still incredibly high, and bringing those down will be vital. Part of that answer, I think, does come down to automation and the ability to reduce the amount of labour that's involved. But I think another part of it that needs to be considered is we've got a new industry being born here. And so industry dynamics, I think, are a really important part of it. You've got a therapeutic company that is producing a therapy that they're potentially going to make billions of dollars on. And you're asking an equipment company potentially to respond to that need and create something without necessarily knowing if that product will ever make it to market, without knowing necessarily what's the requirement of that piece of equipment. And so that's quite tricky in terms of a risk share. How much risk can the equipment company take? How much risk should the equipment company take in developing something to help any one therapeutic company, or a therapeutic company as an industry, if we can start to establish what a universal platform could be? And so I think that's where we've seen challenges from an industry point of view, is we're still seeing the industry start to form and develop as much as the technology. And I think until we see these companies being able to work out how it is we should work together, I think that provides a barrier that we need to work out how we're going to overcome. It won't be one thing. I think that's the thing we can be certain about. I think it's going to be an interplay of a lot of different things. Something that I think the pharmaceutical industry has always struggled with is things like standardization. And the benefit of that is a cost reduction. As we adopt a standard, we can bring down costs. There might be opportunities within cell therapy to think about standardization, whether that's standardization of manufacturing process, so that we've actually got the ability to use a piece of equipment to make multiple therapies on. It might be standardization of the tracking, so that as the therapies arrive in a hospital, clinicians aren't trying to administer multiple different therapies through slightly different tracking routes. It might be standardization through some of the consumables that we use in part of the process, so that the disposable parts of the process have a level of commonality. I think if we look at which parts of the industry there is not a need to competitively differentiate on, I think we can then start to unpick some opportunities to bring costs down, potentially work together to develop common products that help everybody, and ultimately potentially bring those headline costs down to things that are going to make the ability to deploy this much more widely possible. For me, it all comes back to patients again, and what we can do to help patients. And part of that is cost and patient access, but also there's a lot of other pieces to that. So at the moment, you've got the complexity of the patient having the blood taken for the input into the manufacturing process, and then it's not until a few weeks later that the product will be returned to them. That's not necessarily an ideal scenario for a patient. And then you've got geographic challenges. So both in terms of therapy availability, that certain therapies are only available in certain countries, but sometimes you may even have to travel to the one facility that is able to manufacture and administer that therapy. So for me, I think a future therapy manufacturing approach that really looks at how we better serve that. And I think some of that may be around near patient manufacturing. I'm not sure that's the universal solution, but I think for certain therapies, being able to manufacture it near the patient will be very beneficial. I think that also then starts to talk to things like the time to manufacture. Wouldn't it be wonderful if rather than 2 weeks, we could be manufacturing these in a day or maybe a few hours? And that then also talks about things like potency. Which of course is interlinked with cost, because the more potent we can make the therapy, the less time it's necessarily going to take to manufacture it, because we won't need to expand so much, but also the less volume of cells we're going to need to infuse into the patient. So if one day we could have a near patient manufacturing capability that could make the product in an hour, and that we only need maybe 50 millilitres of product to infuse into the patient, that would be my sort of 10-year dream of what this might be. And in a way, part of me thinks that's unlikely and that's optimistic, but if there's one thing I think this industry has taught me, is I've constantly been surprised, particularly been surprised by how far ahead the biology has managed to keep pushing and pushing. So it wouldn't, in many ways, surprise me if we don't see someone making those sorts of breakthroughs in the next few years that could enable that kind of future. I think disease indications is going to be a massive part of the next few years. I think we've seen these trials running over the last few years. I think in a way, I've been a little surprised, and perhaps of the last 12 months have been a big hit on this. I think the last 12 months would have been incredible for cell therapy if it hadn't been for the pandemic. We know that's caused lots of problems with things like recruitment for trials. It's been really, really tough for people. So I'm really hopeful that as the brakes come off post-pandemic, we can get back on to running those trials, that we'll actually start to see new indications coming through. I think hard tumours are absolutely up there. So with what we've done for blood cancers, which has been incredible, the reality is they're still a relatively small indication. if we could start to go for lung cancers, for breast cancers, for solid tumours that we know at the moment for so many of those diseases, the treatments are not that effective. I think that for me is the great hope of the next few years, is bringing those indications on. That will throw up new manufacturing challenges. We know with solid tumours, some of those processes look quite different to an existing blood cancer process, to existing CAR T process. So I think there we're really going to have to see the manufacturing, the equipment really step up. I think we've had our sort of time to prepare for that over the last few years. I think we've started to open the taps a bit on what we can do. My hope is that now in 2021, 22, 23, that all of that starts to come together, that we push ahead, we start to go after new indications. I think that'll be a really exciting time for us as we start to see those new therapies rolled out.